finally looking at the third line of defense. We're looking at the activation of T cells and B cells. So today's video is all about T cells and their response. And it forms this thing called cell-mediated immunity, which are cells killing cells. T cells are going to kill invaders that are living inside of your cells or faulty cells. The third line of defense is all about forming a really specific response and hopefully also getting some long-term memory of this response as well. So when we're looking at memory, it means that the next time this same pathogen gets into your system, we remember it, we know exactly what to do, and we can generate a really fast and big immune response to sort of wipe it out before you even realize that you've got the infection. T cells are an example of a lymphocyte. So lymphocytes are the cells of your lymphatic system. Your T cells are going to mature in your thymus, hence the name T cells, and they hang out in your lymph nodes. The role of T cells is to stimulate and also activate other cells, and we also have some T cells which are responsible for directly killing cells as well. T cells are a bit of like an intermediate step that we have in the third line of defense. Like the overall goal with the third line of defense is to produce our antibodies, which are made by B cells. So T cells are needed to help to activate the B cells. T cells alone though are able to kill certain cells. So if your cell has been infected by a virus or your cell is now a cancer cell, our T cells can kill those cells. They can't really deal with pathogens that live outside of the cell. There are lots of different types of T cells. We've got helper T cells, we've got cytotoxic T cells, which are also known as killer T cells, and we have memory T cells and suppressor T cells. We are going to focus a bit more on the helper T cells and cytotoxic T cells today. Whilst T cells are kind of hanging out in your lymph nodes, they're not active. They're considered naive T cells when they're hanging out in your lymph nodes. We do need to activate them to sort of begin the T cell response. What we need to be able to do is we need an antigen presenting cell. This is our macrophage or our dendritic cell from the second line of defense. These antigen presenting cells have eaten up the pathogen and they've ripped off the antigens from the pathogen. They wear these antigens on what we call major histocompatibility complex or MHC, but they've got class 2 MHCs on their cells. So their cells have MHC class 2 and they're holding an antigen from a pathogen. Once these antigen presenting cells make their way into the lymphatic system, they're moving through lymph nodes and they're hoping to find the one. They're hoping to find the one true T cell to rule them all. Each T cell is going to have a unique receptor, a unique thing called a T cell receptor on their cell surface that is going to bind to the exact right combination of shape. And that shape is the MHC and antigen in combination. The shape that we get from an antigen being held on an MHC class 2 is what we call an epitope. An epitope is like a general term for like a complementary shape to a receptor. So we've got our T cell receptor from our naive T cells, and it will have a complementary shape to the epitope from our antigen and MHC. This is also why it takes a little bit of time to activate T cells. It could take about like 3 to 4 days after an infection to activate the T cell, because we're waiting for these antigen presenting cells to move their way through our lymphatic system to find the exact right T cell to activate. So when the antigen presenting cell finds the exact T cell that it wants to activate, it's going to bind to that T cell and it triggers a chain reaction inside of that T cell. So inside of that T cell, we're going to get a signal to divide, and we're going to get signals in those divided cells to differentiate. Essentially what we're making is a T cell army. We've gotten that one naive T cell activated. It's going to clone itself through mitosis, and these clones are going to start to differentiate. So that means that we're going to switch certain genes on and off inside of that cell to make that cell really specific and give it a particular job. So in this T cell army, we might have more helper T cells being made, we might have some killer T cells being activated, we might have some memory T cells being made as well, and then finally we've got some suppressor T cells. If you've got a helper T cell that has been activated now, these helper T cells will go off through the lymphatic system once more, where they try to find the right B cell to activate in our humoral response. Future video to come. Other helper T cells will move with our cytotoxic T cells to the area of infection. Once they get there, they're going to stimulate all those phagocytes that are working really hard to essentially work harder. And they can also help our cytotoxic T cells do their thing. They're a regulatory cell. They're going to stimulate by releasing chemicals or cytokines, telling people to like, do this, do this, do this. They're a manager. Our cytotoxic T cells are effectors. So their job is to find any cell that has the antigen that it is complementary to on its surface. So this is how we get a really specific response. Our cytotoxic T cells will only kill cells that have this antigen. They're not going to kill healthy cells like we might get with other white blood cells in the second line of defense. Instead, they're going around and checking to make sure they only kill cells that are classified as non-self with this particular antigen. Cytotoxic T cells are only going to directly kill cells that display the antigen that we're looking for. So once they bind on to that infected cell, they're going to release out a couple of different things. They might release out this protein called perforin, which punches holes in the membrane of the cell, and that causes the cell to break open and lyse. They can also release out some granzymes, so similar to like granulocytes, they'll release out these proteins which break down cells from the outside in. Our cytotoxic T cells can also induce apoptosis by binding to the death receptor of cells and triggering death that way. So this is really useful in cancer cells because a lot of cancer cells aren't responding to intrinsic signals, so signals from within to die, and so the only way to sort of get rid of those cancer cells is to stimulate a different pathway for death, which is a signal from outside of the cell. Let's have a look at a cancer cell actually being attacked by a cytotoxic T cell. So you can see it's binding to that cancer cell, and you can see the changes in that cancer cell, and it breaks down and actually starts to trigger the death of the cell. So we are able to get rid of cancer cells using cytotoxic T cells. Cytotoxic T cells, because they do look at intracellular threats, they can also recognize non-self cells such as those from organ transplants as foreign and attack them. In the case of transplanted tissue, we might have MHC1 that is a bit too different from our normal MHC. So we're going to trigger the antigen presenting cells that go and activate our T cells, and we make cytotoxic T cells that will only affect these transplanted tissues. When the cytotoxic T cells get to the area where the transplanted tissue is found, it will trigger apoptosis of these cells and kill this tissue pretty quickly. The other T cells that we have kind of work a little bit after an infection. So once we're done with the infection, those cytotoxic T cells have come in, they've wiped out all of the infection, we have a surplus of T cells. So suppressor T cells will actually induce apoptosis, or the cell death of these extra white blood cells. They'll release out a chemical that kills these cells and gets rid of them, because keeping them around is going to be a bit of a waste of resources. Suppressor T cells will also dampen down the effects of phagocytes, because phagocytes, whilst they're good, sometimes they can damage normal cells as well. Phagocytes are also releasing out a bunch of cytokines, which are triggering lots of different responses, and your immune system can become overworked. When this happens, we might get this thing called a cytokine storm, which causes a lot of really severe symptoms because your immune system's just like, I don't know what's happening, let's just try everything at once. And that is going to really, really deplete your resources. Cytokine storms are found in a lot of different diseases. For example, COVID-19 does induce a cytokine storm in some people, and that's when you get this really severe collapse of the immune system, and it helps the pathogen to actually spread further. As part of the clonal expansion of our T cell army, we did also make some memory T cells. Memory T cells are responsible for sort of hanging back in the lymph nodes, and they will be activated the next time you encounter this pathogen once more. Our memory cells will actually last in your body for a really long time. Some can last for like over 20 years. So that means that in your body, you hold the information for any past infection that you've had that's made its way past that second line of defense. As soon as you've activated those T cells and then eventually activated those B cells, you create these memory cells which will stay in your system for as long as we can. You have information from a lot of infections. So if you encounter the same chickenpox strain that you got when you were 8, you are not going to develop it later on because you have that memory and you can get rid of that virus before it takes hold. If a virus does mutate, however, you've got to start all over again. We've got to start this whole process from scratch. So things like influenza, which mutate really quickly, are going to generate a brand new immune response each and every time you encounter it. So I hope you've learned a bit about T cells and their role in the third line of defense. They are essential in cell-mediated immunity. However, with cell-mediated immunity, it is limited in only working against intracellular threats. If a pathogen has been released from a cell or is not actually an intracellular threat, we do need to call in B cells and antibodies. So happy studying, and I'll see you next time.